and he loved the facts, and he also loved the T cell for one particular thing. So this is really his passion. And after he graduated summa cum laude, he went to Klaus Ley, and together with Klaus, he set up the single cell RNA sequencing and paid a lot of attention to the T cells. He is a bioinformatician specialist now. And I think since a year, he now moved back to Germany. So he started as a junior professor at University of Cologne, and he started his own lab. So I'm very proud of Holger. And Holger, I'm very curious about your T cells and atherosclerosis. So the screen is yours.
Esther, thank you very much for that more than flattering introduction. I'm blushing. So thanks a lot for inviting me to all four of you. And I'm happy to share some a few published data sets, and let's see that I get the techniques here to work, and some unpublished data. Okay. Can you see my presentation? Does everything look okay? All right, great. So let's get started. I don't have to tell this audience basically about the death rate of cardiovascular disease, and that it is still the one of the most causing deaths or leading causes of death in the world. And I just wanted to share some clinical trial data here with you that shows that there is in patients, despite their lipid lowering, or they are managed on lipids, there is a high residual inflammatory risk. And how do we define that, or where do we come up with this? So you see here in the Jupiter trial that the administration of statins reduced the death toll of cardiovascular disease significantly by reducing lipids, but there is still a huge residual inflammatory risk or death toll that we see. And newer therapies on top of standard therapy, evolocumab or other PCSK9 inhibitors, even more reduce the death toll, but there's still a remaining high incidence of cardiovascular disease to cause deaths. And we think of this as the inflammatory risk, which is defined by the immune system that was once triggered by hyperlipidemia, hypercholesterolemia, and the overall atherosclerotic environment that kind of then took an auto turn. So in the early days of atherosclerosis research, it was already clear that it is a chronic autoinflammatory disease or a chronic inflammatory disease. And then under Russell Ross, this kind of idea had a renaissance. And we know that the dysfunctional endothelium by now is activated. There's lipids trapped in the subendothelial space. Chemokines are sensed out, adhesion molecules occur, and immune cells get recruited to the growing atherosclerotic lesion, which really grows in size. We see necrotic core formation. Basically, all immune cells that we know in the body are participating in that immune response, but that's a different talk that I'm not going to focus on today, on the diversity of leukocytes. But I rather want to study or focus today on CD4 T cells. And we think these are some key players in the immune system that we can manipulate to actually help and providing some aid here and stop the immune system from going, so to say, over the cliff in atherosclerosis and prevent that happening. We need to figure out where we can actually push the brakes. And I think these T cells are an important key player to really work against atherosclerosis. So early work from Göran Hansen, actually published 1 year after I was born, shows here that CD4 T cells actually reside in quite significant numbers in the shoulder region of atherosclerotic plaques. And we know that CD4 T cells are actually derived from bone marrow progenitor cells that then home into the thymus. And in the thymus, they will undergo a process of positive and negative selection, rearrange the alpha and beta genes of the TCR, get a unique TCR, and that TCR is then tested in the thymus. And if you're looking at a CD4 T cell, it is restricted against class 2 or MHC class 2, and a CD8 T cell is restricted by class 1. These class 1 or class 2 molecules present antigen to the T cells that they will specifically recognize in a unique TCR combination on their surface. But that is not enough for an activation of a T cell, and ESSA provided a lot of work in atherosclerosis that we need on top of a TCR stimulation, the appropriate amount of co-stimulation. We know that there are pro-atherogenic or pro-inflammatory diets, and they're anti-atherogenic or co-inhibitory diets in atherosclerosis. The integration of just a short glimpse of what the immune system used to modulate antigen-presenting cells and T cells is shown here. There's so many more interactions known, and it's very complicated. Once a T cell, though, has left the thymus, it is called a naive T cell. And a naive T cell is also defined as a T cell that is not antigen experienced yet. And in this slide that I have adapted from a presentation from Annie Lichtman, I really liked it in a way because it shows you how these naive T cells can change their phenotype into an effector cell and who do they work on. And I wanted to spend a quick minute on introducing to you that the naive T cell, depending on the cytokine environment, can either differentiate in a Th1 cell, a Th2 cell, or a Th17 cell, or a TFH cell. So Th1 cell is actually working on macrophages and causes the classical M1 macrophage to be activated and aids in killing intracellular pathogens. Th2 cells are more known to play a role in allergy, but overall they activate eosinophils by their cytokines and also aid in M2 activation. We usually need them to defend ourselves against helminths and other little worms. Th17 cells activate neutrophils. They cause neutrophil recruitment and activation in defense of the body against extracellular bacteria and fungi. And last but not least, T follicular helper cells, a cell type that you really do find only in secondary lymphoid organs, or in vast majority, where they talk to the B cells and have the help the B cells to produce antibodies and also hypermutate them and produce very specific antibodies that are targeting particularly pathogens. They all have a role in disease as well, not only in fighting off bacteria or pathogens, but also contribute to autoimmunity and allergy. And we know that all of these cells actually play a role in atherosclerosis as well, and we lately summarized that in a review here. And it looks quite complicated, but what I want to show you actually is that the 4 T cell subsets that we know from a lot of studies where we knocked out, or the people in the field knocked out hallmark cytokines or hallmark transcription factor, reveal that Th1 cells, by the production of interferon gamma and TNF alpha, really drive inflammation in atherosclerosis. And regulatory T cells, shown here on the right, by the production of anti-inflammatory cytokines, really contribute an anti-inflammatory response in atherosclerosis and really seem to suppress the immune system in atherosclerosis. And hold that thought for later, because it does not seem to be as simple as we indicate already here. We think that there's actually something more to these Tregs that in atherosclerosis become dysfunctional and take on fates that they're not supposed to have, and thus actually contribute to atherosclerosis in a detrimental way. So where does actually idea come from that we find CD4 T cells that are in lesions and that really do something in there? So Göran Hansson then followed up with a lot of seminal work, really delineating that we have activated CD4 T cells in human atherosclerotic lesions. And he also could show that they are oligoclonal. That is always a sign of an active CD4 T cell response, because the T cells will proliferate if they see cognate antigen. That is highly specific. And if we see, let's say 500 T cells, And among those T cells, already 40 would have the same T cell receptor. We can talk about an expansion of these 40 cells that responded to that particular antigen. And I show you some data from Johan Hansen here from 1995, where they took T cells from human atherosclerotic plaques and stimulated them with OxLDL and measured proliferation. You can nicely see here that these 3 clones obtained from human atherosclerotic plaques really responded to a restimulation with OxLDL, and some also did to native LDL, whereas also some responded to control peptides. Work in Klaus's lab also a bunch of years ago shows that this is happening in experimental atherosclerosis. And this essay from Katja Koltsova at that time, you can see actually by this nice imaging strategy here that the CD4 T cells are labeled in red and the dendritic cells are labeled in green. You see it nicely here that in atherosclerotic lesions in that co-culture system where the dendritic cells are green, T cells reside close to these greenish dendritic cells labeled in red. And the interesting observation that Katja made was that when you use plaque 6-derived CD4 T cells, they are not really so much interacting with the dendritic cells. They go into the lesion, but they're not closely residing to the dendritic cells. But CD4 T cells from ApoE deficient mice that are hypercholesterolemic and have atherosclerosis really show a nice interaction in the aorta and reside next to the dendritic cells. So that kind of sparked the idea in Klaus's lab that there must be an antigen-specific interaction of CD4 T cells. So one of the first things that were done in Klaus's lab back in the day was actually a tiling approach of, I show you only the human ApoB peptides here, but we did the same also for mouse peptides. So what basically was done, then we used a supercomputer and generated 15 mer peptides of the entire linearized structure of ApoB-100, and then tested in a computer-based algorithm how these peptides are binding towards human MHC class 2 variants. There is a nice online tool at the La Jolla Institute. It's the Human Epitope database, where collaborations were really of utmost help to get to this data here. And you see basically across these different human MHC class 2 variants, and humans are quite more complex than mice. Mice lab strains have only one MHC class 2 allele. Humans have many, up to 126. We see that actually some peptides are really poor binders, depicted here in white, and some are really broadly binding to almost all the MHC class 2 variants that were tested. So these are fantastic candidates to study the immune response. We can probe different humans, and I will get back to this in a little later. But basically, these kind of assays were used to define for humans and mice peptides that are likely to bind in a computer-based assay, and were verified later on an in vitro assay where a peptide known with a known binding affinity to class 2 that was radioactively marked was used in this displacement assay. So some of these are really great candidates for something that we think is the endeavor to designing a vaccine in atherosclerosis, and I will touch on this a little later. So with the mouse data, we designed a tetramer. So a tetramer is basically shown here on the left, at least in this cartoon, consists of 4 different MHC class 2 variants that will work in the mouse. MHC class 2 in black 6 is IAB. It's labeled with APC or PE as fluorochromes and loaded with a mouse ApoB peptide. And now in this case here, we use P6. We have also worked with P18, and I'll show you that this works as well later. And then you can use these to fish out in a pool of T cells, these T cells with a cognate TCR, or in other words, T cells that will respond to ApoB peptide in MHC class 2. You see nicely here that in a wild-type mouse, most of the ApoB T cells are actually naive, but compared to an ApoB-negative cells, there's certainly more central memory cells and effector memory cells already in a naive black 6 mouse. And that changes even more so in an ApoE deficient mouse at the age of 8 weeks, where we do not really see atherosclerosis yet, but we have hypercholesterolemia just starting. We see that there's way more central memory and effector memory T cells among those cells that respond to the tetramer. That was already very interesting because it kind of pointed us towards an enhanced activation of T cells already in wild-type mice, which was unusual because you see here, we would actually think that lab mice are not really exposed to a lot of antigens and they're relatively clean. And now there's a huge response towards ApoB, let's say, on a central memory and effector memory level. So we further then looked at hallmark transcription factors by flow cytometry, which I introduced before, and TBAT then marking Th1 cells, raw gamma T marking Th17 cells, and FOXP3 marking basically regulatory T cells. And you can nicely see here that in the ApoB-negative cells, so in all CD4 T cells that are CD44 positive, that are activated or have seen antigen at least once in their lifetime, we have a lot of regulatory cells, and that is kind of expected. That is what we would expect. We have 20% in the CD44 or 5% in the CD44 negatives, and we know that Tregs are a little bit more activated. They need to see antigen once in a while in the tissue where they're residing. So we see a little bit more FOXP3 positive cells in the ApoB negative cells. This though completely shifted in the ApoB positive cells. They were almost non-positive for FOXP3 anymore, or we had at least a drastic reduction from 32 to 70% or even down to 1%. There was a huge expansion now of the raw gamma T expressors. And note that the FOXP3 expressors here double express also raw gamma T. This is also quantified in this bar graph here. You basically see in mice that are 8 weeks of age or 20 weeks of age, in this case ApoE mice, that the polyclonal CD4 T cells are mostly Tregs and some other phenotypes. These double phenotype though does not occur here, but in atherosclerosis we see really that these FOXP3 raw gamma T double positive cells are coming up, pointing that the Tregs here obtain a completely different feature that are responding to ApoB. And we sorted those cells and then performed bulk RNA sequencing of ApoB negative cells or non-regulatory T cells, ApoB positive cells, and Tregs. And you see nicely here in these heat maps, the Tregs really enrich for Treg genes as we would expect. The non-Tregs enrich for genes for non-Tregs like Th17, Th1, some Th2, and T follicular helper cell genes. But the ApoB positive cells are rarely enriching for the Treg genes, but more so having a mixed signature of Th1 hallmark genes, Th17, Th2, and T follicular helper cell genes. So that kind of pointed us that the ApoB-reactive CD4 T cells in this atherosclerosis mouse model are completely different than what we actually know about the CD4 T cells in general that are protecting us from pathogens, fungi, extracellular bacteria. We also performed a single-cell RNA sequencing experiment here where we used ApoB negative cells and ApoB positive cells. We sorted them again by the tetramer, and to really dive into individual clusters of cells. And you see really nicely here, the ApoB positive cells are clustering among their own away from the ApoB negative cells, and they really form their own kind of mixed phenotype lineages. And that is something actually that has been seen by Chiara Gianarelli lately as well in human plaque studies where she used site-seq and single-cell RNA sequencing. You see here that the plaque T cells cluster away from the blood T cells, so they're also again very different. And among, as I'm just exemplifying that here, among these plaque cells, there were CD4 T cells very much enriched this cluster. And in patients that were in her paper considered asymptomatic, These had an interferon gamma enriched pathway. They showed IL-12 and also toll-like receptor cascades, so they're a little bit more like classical Th1 cells. But in symptomatic patients, they really obtained a very different functional phenotype. So they are enriched for cell pathways such as Rho GTPases or IL-2 signaling and PDGFR beta. So pointing really that the CD4 T cell responses that we know from classical immunology might not be something that we can just copy one-to-one in atherosclerosis. And we followed them up and really figuring out, are these cells, where are they coming from? Because we saw these FOXP3 positive, ROgamma T positive cells, are they Tregs that are on the way to a transition? We know in the intestine, there are FOXP3, ROgamma T double producing cells that are instrumental in keeping the gut homeostasis and protecting us from certain bacteria. So we used here a fate mapper mouse, and under the control of FOXP3 YFP green, where we have, when cells express FOXP3, always a yellow fluorescent protein, we crossed them to the ROSA26 flox RFP mouse. So that means the offspring is in a current Treg, yellow and red, as indicated here. But if a Treg loses its hallmark transcription factor, it is really implementing the suppressive program and also the lineage identifying program, these cells turn red. And in atherosclerosis and ApoB-negative cells here, you see that roughly 3.1% of the population in atherosclerosis have lost their FOXP3 expression and turned on red, so they are not Tregs anymore, but they have been once Tregs. In ApoB positive cells, that is really doubling. So we really see more Tregs losing their phenotype in atherosclerosis that are responding to ApoB-100 peptides. And you see that quantified here. In the CHO diet, these kind of are almost equally leveled out. So we think it's really something that is diet-induced, that these cells are switching. So to further confirm that, we wanted to address it in a different way. So we use CD45.1 marked Apoe mice, isolated ApoB positive Tregs, again by the tetramer, or ApoB negative Tregs, and subjected them to CD45.2 Apoe deficient mice that were pre-fed a high-fit diet. So pre-transfer, all these cells were actually regulatory T cells. The only difference is that some were ApoB positive and reactive, the others not. You see nicely here in that flow cytometric analysis that more than half of the Tregs 6 weeks post-transfer lost FOXP3 expression. So this high atherosclerotic environment makes these Tregs lose their Tregness and switch. And in that experiment here, we had a look at the outcome in atherosclerosis after we used the same experimental setup as introduced before. And you see really here in a quantification of atherosclerosis that ApoB positive Tregs fail to protect from atherosclerosis. This is something where we're actually expecting a nice protection, and in the ApoB negative Tregs that were polyclonal, not a protection anymore. But here in that setup where we transferred 50,000 cells twice, the lesion size did not differ from control mice that did not receive any cells anymore. Again, pointing towards that the switching regulatory cells, polyclonal as well as ApoB-reactive, are not capable of protecting from atherosclerosis anymore. So that's all nice, but what about humans? Do they have ApoB-reactive T cells, and how do these T cells look like? And I showed you this map already before. We have chosen here in the first set of experiments to top 20 ApoB peptides that are broadly binding to a whole plenty of different MHC alleles. It's very more difficult in humans to use HLA-based tetramers, because we can only address a small fraction of the population. For example, DRB0701 is present in 7 to 15% of the population, which is still quite high, but that means you really are limiting yourselves in the amounts or the number of patients that you can address. So we designed a peptide restimulation assay as shown here, where we isolated PBMCs from healthy controls or from cardiovascular disease patients, and either left them unstimulated or stimulated them with ApoB peptides or with a super antigen where we know that it's causing quite a drastic T cell activation. And intracellular CD4 ligand is a surrogate after 5 hours that faithfully leads or indicates T cell activation. We see already here that cardiovascular disease patients have a 2 to 3-fold expansion of these ApoB-reactive T cells in their circulation compared to the healthy controls. And then we looked for some cytokines that they're producing, and you see that actually here, these cells express way more TNF-alpha compared to cells from healthy controls, and they express also way more IL-17 compared to healthy controls or not stimulated T cells, suggesting that here, indeed, cardiovascular disease patients have ApoB-reactive T cells, and these are in cardiovascular disease more pro-inflammatory than in healthy controls. I did not show the data here, and it's actually very interesting. It's opposite for IL-10. So in healthy controls, we have more IL-10 producers, but in cardiovascular disease patients, there are less IL-10 producer, and IL-10 is a very strong anti-atherosclerotic and anti-inflammatory cytokine. And I introduced to you already the tetramer technology beforehand. We still wanted to survey, though, a little bit more, how do individual T cells respond? And here we used HLA-DRB-0701, and we had a huge patient biobank where we could, where we knew the different HLA types, so we could actually survey and DRB-0701 phenotypes. And we have chosen P18, which is an ApoB peptide that is sequence identical in mice and humans. And that's the only peptide in the entire sequence in mice and humans ApoB that we can utilize here. And I want to show you the gating strategy next. And you see really these tetramer works nicely by including all the non-T cells and just gating on a CD4 positive T cells. You see in a DRB or 701 positive population, quite some T cells that are bound or binding APC P18 loaded tetramer or PE P18 loaded tetramer, whereas there's almost no T cells in the mismatch control that respond to this TCR. So that is a very, very nice tool to study T cell responses, at least in a DRB1 or 701 patient population. And that database was further, or this biobank was further great because it is the WISE cohort. So they actually also performed ultrasound on the carotids. So we knew which patients that were included had actually subclinical atherosclerosis or atherosclerotic lesions, and which patient didn't. So we only had the chance to get hands on a very limited amount of samples due to the limitations that I just mentioned. We saw a trend towards more P18 specific cells in cardiovascular disease patients, so suggesting they're expanded. But what we found was really striking and very, very interesting. What I've shown you in our atherosclerosis mouse data is, and don't worry too much about these different pie charts, that looks very complicated, but just focus on this orange slice here, which is nicely expanding. We have a dramatic increase in the number of FOXP3 positive, ROR-gamma T positive cells, as we saw in the mice. So we think that in patients with subclinical cardiovascular disease, again, these cells are expanding and obtaining a different, more pro-inflammatory phenotype. So the question is, of course, how do different patient consortia look like? How do people with comorbidities or how are their T cell responses actually looking like? And that is something that Lauren DeVore in my lab is going to study in the next couple of years. And she will look at the phenotype of the ApoB-reactive T cells in populations with high cardiovascular risk. And we established a collaboration here, and we're going to sample material from patients with obesity and also undergoing a subsequent intervention program to see if the phenotype is reversible. So to summarize this part, we have I hopefully could convince you that there are ApoB-reactive T cells in healthy and cardiovascular diseased individuals, and their phenotype changes from a regulatory to pro-inflammatory phenotypes in cardiovascular disease patients and atherosclerotic mice. But why do we actually care about CD4 T cells that respond towards ApoB in an antigen-specific manner? And we think they are a fantastic new way to actually treat atherosclerosis. And antigen-specific T cells are key in a vaccination-based approach. Why is that? Because they have a unique TCR that responds to unique peptides. So you can almost surgically intervene in your immune system and really activate only certain particular kinds of T cells to respond. All the T cells in the vaccination that are not responded to an antigen are not really affected by the vaccination. And we have chosen in that particular experiment here the P18 peptide of ApoB-100, which is sequence identical between mice and humans. And you see in that nice experiment, we were really happy to actually see that reduction in atherosclerosis in mice that received the P18 peptide with two kinds of adjuvants, which you need to wake up the immune system and to mount a full immune response towards just adjuvant controls. And this vaccination was atheroprotective by expanding also P18 specific T cells in the spleen, but also in lymph nodes. We saw, you can see this here in these flow cytometry plots, and here it's quantified. There's really way more P18 antigen specific T cells after vaccination. And strikingly, these P18 expanded T cells in the vaccination setting are also more regulatory. They have more FOXP, or they express FOXP3, and we can almost see a doubling to tripling of regulatory T cells that are antigen specific. So that's why we think we have a reduction in atherosclerosis. So that was really great, but if you think of it in a way, what are the phenotypes of the other expanding T cells? Almost 80% of the expanded cells are actually not FOXP3 positive cells. So that got us a little bit worried, and we have to, to translate an atherosclerosis vaccine to figure out what is really going on here, that we are not building up a therapy that is in the long run actually intuitive and activating the immune system. So we used here, again, a single-cell RNA sequencing experiment of ApoB-reactive T cells. And this time, we chose a slightly different strategy. We did not use, as Dennis has published, a 10x approach, but we used a SMART-seq2 approach for two reasons. 10x genomics is great if you want to look at overall leukocyte diversity or cellular diversity, but it's not really deep. It's relatively shallow. And a lot of the transcription factors and cytokines are really lowly expressed, and you do not fish them out by 10x genomics-based single-cell RNA-seq. SMART-seq, though, gives you the whole transcriptome of a T cell. And on top, it allows you to perform some really nice bioinformatic workflow, which Janal in Klaus's lab performed. And this is the workflow that we show here. I don't want to go too much into the detail, but he used basically an algorithm called Tracer developed in the lab of Sarah Teichmann. And Tracer gives you the opportunity to do a de novo TCR assembly in your sequencing data. So you get both in the same assay, a single cell transcriptome and the TCR expression of the CD4 T cell at the same time. And that allows you to actually survey clonality of these T cells. And here in these clonality plots, you see the usage of the alpha V and J chain usage and the beta V and J chain usage. And what you see is actually that among the 32 first clones that we had a look at from one mouse, more than half of the cells express the same alpha chain, the same J segment, the same beta chain, and the same J segment. So these cells are oligoclonally expanded. So again, among 32 cells, more than half of them responded to the P18 vaccination. The other half did not really, and we can later discuss why that might be. We are not so sure yet. But if you cluster now the clonally expanded CD4 T cells against the control T cells that were included in that experiment, and we use CD44 positive T cells here that are not responding to the antigen, you see that they cluster again among themselves. They form, again, a unique population. And now you can layer on top of that map the clonality. And you see that in black, the negative cells are not clonally expanded, and some of the expanded groups are hiding within the negative cells. So they share some transcriptional features with them, but most of the cells that are derived from the P18 vaccination cluster together and share a transcriptome. And we layered now or surveyed these 32 different cells for their expression of hallmark transcriptional cytokines and also, cytokines and transcription factors. And you see here that the majority of the expanded cells have a Treg signature, so they express the hallmark transcription factor FOXP3, they express CTLA4, which is an immunosuppressive co-inhibitory molecule, they express GITR, which is known to be very highly expressed on Tregs, and only to some extent a few genes of other lineages. We have the one population that expanded with the P18 that has a residual regulatory T cell signature. You see some FOXP3 in this cluster 4, but a strong interferon gamma receptor signature and some TBX21, which encodes the TH1 lineage transcription factor TBAP. So that overall means this heat map here that we have expansion of cells, or we mostly see expansion of regulatory T cells by the vaccination. But what we saw by flow cytometry is that we also get other cells to expand, such as cells with TH1 signatures, TH17 signatures, and T follicular helper cell signatures. So the next years experiments will really need to address when we're vaccinating how stable are those cell lines, and are they further expanding in the progression of atherosclerosis to really make sure that we have a safe strategy here to prevent atherosclerosis in the long run. So with this, I'd actually like to conclude and thank everyone that was involved throughout the years. Most of the work that I've been showing you today was from Klaus Lei's lab during my postdoc, and Yanal, Koji, and Dennis Wolf at that time were drivers of these entire projects, and I was very happy to work with them on these very exciting different experiments. William Kwok from the Benaroya Institute provided us with tetramers. We couldn't have done it without him. That addressed human specimen, and Mark Jenkins provided us with mouse tetramers. And then I'd like, of course, to thank my team here at the University of Cologne and the group of the pediatrics department with whom we are going to set up a very nice study. And I would like to thank you for your attention and be happy to take any questions.
Thank you very much, Holger. That was beautiful. Are there any questions for Holger? And please use the raise hand button.
Can I manually raise my hand and ask a question? That was beautiful, Holger. Thank you for updating us with all of the very complex aspects of this T cell biology. But the complexity, I feel like at that last part got so interesting. So you bioinformatically determined the T cell receptor clonality. Is that what you didn't actually sequence the protein, like the actual receptor, you just sequenced the RNA transcripts and then determined and then from there did the subsets of what are they expressing and how am I getting that right? That is amazing.
Absolutely. Yeah, absolutely correct. Well, thanks a lot, Katie, first. And yeah, this is absolutely correct. And this is really, really cool. The software from Sarah Teichmann allows you to really do a de novo TCR assembly from the transcriptome. You have to use, though, and that should be a disclaimer, high quality data that is very deep.
That's what I was going to ask. The sequencing must be very deep, right?
Yeah, so the sequencing must be deep. You need SmartSeq2 or data of a similar quality. The reason is 10x is not good enough, right? The 3' strategy from 10x just gives you a read from the 3' on and leads into nowhereness, so to say. And what you need for the TCR, because it's relatively large, is a sequencing read from both sides coming in, that you really capture the full length TCR. And the specialty about the TCR is, and I did gloss over that in the introduction, there's multiple layers of how the TCR gains complexity. It's not only the VDJ recombination of the alpha gene or the alpha segment and the beta segment and their randomized pairing, but within the region of the CDR3 in the TCR. So the region that the T-cell receptor recognizes antigen, there in enzymes will will without a template introduce nucleotides. So the diversity that is created by that is tremendous. And therefore you need to have these very deep sequencing approaches to really tackle that. From the 32 cells that we had in this first experiment, we had something more than 70% of TCRs retrieved. And that is actually what you can do right now by our technical limitations. You could use the 10x 5' strategy, which really gives you TCR, BCR, and there you will get similar numbers in the recovery of TCR. So not every T cell will give you later in your 10x experiment or in the SmartSeq experiments a VDJ recombined TCR.
Has there been any effort in doing combining this with like a spatial approach within the plaque, presumably, because that's initially where a lot of this was revealed, right? That proximity of the T cells to, a lot of the antigens that, presumably is what, how this is at least partially being activated. Has there been any move, or is this something you're thinking about sort of doing it, like adding on this spatial aspect to it so that those T cells that you're getting aren't necessarily in the circulation, but they're coming from potentially the actual, tissue itself?
Yeah, that's a great idea. And we are thinking a little bit about this. Dennis already had done some initial approaches by clustering transcriptomes from single cell experiments of ApoB positive cells on the plaque cells we have in our leukocyte diversity paper. And they cluster pretty well. They really align very well. But the problem is the tetramer does not really work that nicely on digested aortas. And you have only a few hundred T cells in the plaque, right? Or in the entire inflamed aorta. And then it's just a numbers game. We would not expect that every T cell in the plaque responds to one peptide of ApoB-100, but there will be many responding to maybe ApoB-100 or HSP60, 65, would also be candidate antigens. So that's a little the problem. But we did an experiment where we combined or compared from all CD4 T cells, the TCR beta chain usage, and compared that to the TCR beta chain usage in the lymph node and spleen resident ApoB-reactive T cells, and there's quite some nice overlap. So suggesting these cells might go from the spleen and go to the plaque. But it's a surrogate way to approach it. But we would love to address it, Katie, to really see where are these.
I mean, in a way, the technical limitations are so massive, but yet here you are presenting, deep sequencing on such small number of cells. And it feels like that's just going to be the next step, right? And that's going to be like the tools are going to get stronger and you'll be able to do it. Wonderful. Thank you.
Thank you, Katie.
Yeah. Ada, you had a question too.
Yes. Thanks, Holger. Great presentation. I think we discussed it before, but maybe I will bring it up again about the autoimmunity, right? Because you are immunizing against a native protein that's very abundant in the liver, right? So I wonder if you ever checked for autoimmunity, if there are maybe epitopes of ApoB that are modified, you know, in atherosclerosis, that maybe you can immunize for them that are not supposed to be present in non-atherosclerosis settings.
Yeah, I think that's a fantastic question, Alda. And I mean, overall, we see OxLDL being one of the prime candidates that really triggers that autoimmune-like component, right? We see antibodies against OxLDL, as Christoph Binder lately showed, again, very nicely in the seminar here. And we see T cells that respond to ApoB-100 peptides. I think overall in the oxidation of LDL, it's mostly that the lipids are oxidized, but not so much the protein. The protein stays more or less the protein. What we observe though, and I wanted to show in a way, is really, if you want to bring it down into a nutshell, we have ApoB-reactive Tregs in healthy individuals and healthy mice. And in atherosclerosis, these cells switch to pro-inflammatory phenotype and then cannot really protect us anymore. So we have this gradual loss of protective autoimmunity. I should be very careful. It's not really an autoimmune disease, but autoimmunity. And thereby these cells switching to Th1, Th17 cells to become actually contributors to that disease progression. So we did not observe anything in terms of cholesterol levels or overall leukocyte counts when we vaccinate against ApoB peptides. What we do see though is an antibody response towards these peptides. Absolutely, yeah. But it will get challenging to vaccinate against modified peptides because it's more so the lipid antigens that are modified in atherosclerosis, to my understanding, than the protein antigens. The ApoB will still relatively stay intact, but that's a speculation here, and from what I understand, talking to
yeah, I wonder if like another approach could be, you know, lipoproteins, right? Instead of taking just the ApoB, just maybe looking at reactive T cells to lipoproteins that are maybe increased in atherosclerosis settings. Because then you might have like epitopes of ApoB that are, you know, showing with interactions with lipids, which can maybe lead to other epitopes, new epitopes, right?
Absolutely. And yeah, that's a very well taken point. I think what we overall want to strategize in the long run on is to induce a T cell response that is of regulatory nature and dampening the immune system within the plaque. So just telling ApoB-reactive T cells that are more Th1-like to calm down and to have OxLDL antibodies boosted. And we see that when we vaccinate it, we get more of these antibodies to cover these OxLDL particles and foster either their clearance or even prevent them from being taken up, right? Depending on the stage. Absolutely. And it's all a matter of, and that's why I think the project of Lauren is going to be very exciting, because you need to know at which point is which population at risk overall. You would not like to vaccinate a patient or that is relatively old and has already pronounced atherosclerosis, because we might, exactly because of that switch, induce a pathogenic T cell response. There's one paper out there where they looked at this phenomenon, and one peptide did not change atherosclerosis at all, but a different peptide vaccinated when atherosclerosis is already established really boosts a pro-inflammatory response, and these mice actually got more atherosclerosis. So that is also an open question we need to address in the years to come. Antigen, adjuvants, timing, yeah, absolutely. Great, yeah, great point.
Thank you.
Oh, Jesse, you're next.
Thank you. Holger, actually, it was good to hear that you've had trouble with tetramers and plaques, because we tried those also in the LDL model, and we can get the same like systemic pools of these cells, but I never could get the plaque cells to stain for the tetramer. So I'm actually glad to hear you're having the same problems. That's not my question. So actually, we're interested in trafficking of myeloid cells, and you did that cool but very difficult transfer experiment. Did you do trafficking analysis of the different Treg phenotypes and their ability to move through the tissues or enter relevant tissues like spleen or even the plaque?
Yeah, I think we have that data in the manuscript. We certainly did flow cytometry on the aortas, and in the aortas, you see that most of the, I gotta say, it's a tiny amount of cells that finally from a systemic injection will end up in the atherosclerotic plaque, but most of them switched from Tregs to being, well, I think 60–70% are not Tregs anymore, 6 weeks post-transfer. So we did these experiments in the spleen. I cannot honestly remember the data, how they were switching. You find
that switch, is that switch enabling them to now enter, or is it that once they get in, I guess it's a weird question, but
no, I think it's a valid question, and I cannot really address it. What I find very interesting, though, is work from Elena Galkina and Klaus a few years ago, where they see the CCR5 interferon gamma producing Tregs that are established in advanced atherosclerosis. And we know that CCR5 is a main driving force to get T cells and also other immune cells into the plaque. And an interesting thing about these interferon gamma producing Tregs is that they still suppress cytokine production by other T cells, but they do not suppress their proliferation anymore. So they're kind of somewhat losing their suppressive capacities, but not totally yet. You see them, if I remember correctly, 6 months after Western diet. So it takes quite some while, but then you see those cells that are having a different fate in atherosclerosis. And for the tetramer, yeah, we eventually gave up. I just say it like it is, right? I mean, I think what is very different in the CD4 tetramers versus the CD8 tetramers is that the enzymatic digestion of the aorta really affects the CD4 expression. It does not so much affect the CD8 expression. I think the CD4 is necessary to bind to the tetramer to pull out the T cells and have the tetramer sticking to it. And if you really lose the CD4, and even if there might be T cells that would respond to the tetramer, it's hard to get them out. Yeah, absolutely.
Okay, Norbert, the dream team is complete again. So.
Holger, great talk, and nice to see you. I have a question regarding these. So when we look at human studies about T cells and their predictive value in cardiovascular disease, and in particular acute myocardial infarction, there's a cell population which is CD28 negative, that is highly predictive of cardiovascular disease and adverse outcomes. Is that somehow related to the population you see, or have you looked in your population in either the tetramer positive ones in general, or in particular those that are kind of like the X-Tregs that are Th1 or Th17 like? Have you looked at CD28 expression in these? Are they negative for CD28?
So we have not looked at that, and it's a great question. We are setting this up right now in our SciTech panels. So we want to actually also look at these cells. The thing is that the mouse doesn't have CD28 negative cells. These CD28 star cells are only present in humans. We don't find them in the mouse, so we can unfortunately not look into the phenotype of these cells in mice. But yeah, you're right. I mean, we don't know if these CD28 star cells are among the tetramer positive cells in human populations. Restimulation assays are a little bit more tricky to look at because these cells are cultured for up to 14 days in a petri dish to really expand them to get enough numbers. And then some of these markers will not get faithful anymore. So we don't know what we see in a petri dish is actually what we see in the patient when we do the restimulation assay. And with the tetramers, we are limited to 7 to 15% of the population of all patients that we could survey. So that is kind of the two problems that we're looking at right now. But
I didn't fully get that. Do you see these tetramer positive cells circulating also a lot? Because you showed mostly stuff, I think, in the plaque, but you see also some of them in the circulation, right? So in your human data.
In the human data, so we have not looked into the plaque. Only Jörgen looked into the plaque, and I think there was another paper 2-3 years ago from a group in Sweden. I'm sorry that I forgot the name, but they also looked at T-cell responses in the plaque to a different antigen. We have not actually studied that. We only looked in the circulation of healthy volunteers and of cardiovascular disease patients. Okay. But in the first WISE cohort study, we had only up to 20 patients that were a tetramer, or they would suit the tetramer that we have. Not all tetramers work equally well. Some are sticky. There's a lot of technical problems with these tetramers. Mark Jenkins produces mouse tetramers that have a stable expression of the peptide. The tetramers that we got from Bill Kwok do not have that feature. So these assays get so much more complicated because the tetramers are something that you need to use within a couple of months. Otherwise, the peptide will, diffuse out of the binding growth of the class 2 again. And with a restimulation assay, you see, or you can really address more patients. And that is something that is happening in Klaus's lab right now. They're expanding, of course, all these studies on way bigger cohorts of patients. And with a restimulation assay, you can only address then some markers again, or some cytokines, but not all again. So that's kind of the trade-offs that we're facing here.
All right, thanks.
Okay,
thank you,
Robert. Then I'll post the final question because I think it's already 7 o'clock here. Holger, what do you expect from your new studies? Do you think, because you also mentioned obese patients, patients with a lot of comorbidities, do you think the image will be as clear as in atherosclerosis, that you have differences in these ApoB Tregs and these, because also if you look at obesity, maybe that's not so ApoB oriented. And what do you expect there? And are you looking for a diverse pattern of even more specific T cells?
Yeah, absolutely. That's a great question. So we are actually wondering ourselves what we should expect from that study, and we want to really age classify those obese patients, and we're also looking to survey kids that are 8 to 12 years old, because at that point, from what I hear or learn from the pediatricians, the metabolic switch, this hypertriglyceridemia and hypercholesterolemia, is not happening yet. And we think that hypercholesterolemia is one of the main driving forces that switches, or is one of the main contributors to the switch of the Tregs. So it would be tempting to speculate at this point that the kids that are obese, that have not had these metabolic alterations yet, have normal Tregs that respond to ApoB-100, and the ones that did already and face these metabolic transitions, and also adults with obesity will have these changes. But that has to be proven. That's just a wild hypothesis as of now.
Yeah, So, well, thanks, Holger, for your beautiful presentation and also for all your answers to the questions. I thank everyone for listening in. And if you want to chat with Holger or get some advice from Holger, please stick around, and maybe Ada or Mabruka want to take over, and then we'll make you co-host, or you already are. And would say have a nice weekend.
Thank you.
Have a nice weekend. Thanks for having me. It was really fun. It was good. Very lively discussion.
Yeah, I mean, I have like 1,000 more questions for you if you want.
Absolutely. Go for it. Hope I find some answers.
I wonder why you're using ApoE knockouts for your mouse studies.
Over LDLR or?
Yeah, because I think like the LDL particle itself would be a little different in ApoE knockouts, which would not be in LDLRs. Right, because the ApoE is one of the major components of the ApoB particles.
Isn't it more like an uptaker though? And the circulating LDL cholesterol itself is more apolipoprotein B100, and ApoE is an acceptor, the way I understand it, but maybe.
Yeah, I mean.
So it has more to do with the uptake of the LDL cholesterol, right? Isn't it the case? And I gotta say, this is weak terrain for me. I'm not so good with these lipoproteins and how they are assembled and what is in them.
I think ApoE is on the ApoB particles, and it's like one of the major target for the LDL receptor or other receptors that uptake lipoproteins.
Okay. Gotta say, I'm blanking on that. I thought always ApoE is different from LDL, and ApoB is in the LDL cholesterol, to be honest. But it's a good question. I should really read up on this and look into that again. So we chose the ApoE mice because they developed this mild hypercholesterolemia over time. And that was tempting to have these two approaches right away. We either have a mild phenotype or we feed them this Western type diet. We have a pronounced phenotype, and we can study both simultaneously, right, compared to normal black 6 mice. But yeah, I would love to hear your thoughts on that. What do you think? What is going to happen if we, I mean, the LDLR mice were something we used only in in 2 studies, I think. Yeah. And there we saw also protection. But what does it mean in the end? We didn't really, so we only had an outcome study. We didn't really use the tetramers in there. Mm-hmm. Yeah.
Yeah, just something to consider, I guess, for the
future.
Absolutely.
Yeah, it's a very valid point. I always felt like ApoE is more the acceptor and ApoB is giving structure to the LDL cholesterol particle. And I mean, we still have LDL cholesterol if you knock out ApoE, right? It's more that you accumulate LDL cholesterol. Mm-hmm. It's higher, but it's not really cleared anymore. So I thought, oh, it's like it attaches to it.
I think there is ApoE on the LDL particles, right? Mm. And what does it mean when you don't have ApoE anymore?
Right.
I'm not sure. In terms of T cell responses, right? And I mean, I think also you raised a really nice point at the end where you were talking about the children.
Sorry, Ada, you're cutting a little bit.
Can you hear me?
Can you maybe repeat it?
My internet is not very stable.
I was worried it's my internet.
Am I now cutting? No.
Yeah. So can I go ahead with my questions while she's come back?
Absolutely.
Yeah,
I think it's my internet.
She's back. You're back? Ada? Okay.
Just for a hot second.
So I like the graph, the pie chart that you showed in your presentation.
No, go ahead.
So the pie chart that you showed with cardiovascular disease and non-cardiovascular disease, and you saw the expansion of the population of T cells. So those people are cardiovascular disease people. What kind of disease do they have? Do you have any information about them? It's like how advanced their atherosclerosis is? Do they have hypertension or not? Are they diabetic or not?
Yeah, we do. I cannot tell you from, so we excluded diabetics or comorbidities, but it was a first study. So we had to start somewhere and looking into patients. It's not an extensive study, and we should disclaim at this point, the WISE cohort is the Women's Health in HIV Interagency Study. So basically, we have patients in there with HIV, patients with HIV and cardiovascular disease, and we couldn't be too picky at that point, and had like a few of them cardiovascular disease patients, also HIV infected. That's of course a huge co-founder. We could not exclude it at that point though, because we did have only so few samples that we could address.
That's cool.
Overall comorbidities are something that we're trying to rule out now here, and also in a way larger study that is executed in Klaus's lab right now, where they look at the MESA study, the huge atherosclerosis study, and they got shipped several hundred samples. So I think we will certainly in the next couple of years see a really more detailed and more complete picture of what's going on with the immune cells. But it's an absolutely fair question. The peptide restimulation assays that were done in the other paper, that were patients that were really in the catheter lab. So they had severe coronary heart disease incidence. Yeah.
So this is my follow-up questions, is that if they have severe myocardial infarctions or they have plaque rupture, what's your expectation to see in the shift in populations of T cells? What expectation?
Yeah, it's a great question. At this point, I can also again just wave my hands a lot, right? Because we still don't know if it's hen or egg. We know that there's switching in atherosclerosis. We know that they lose their regulatory phenotype if they're ApoB specific, but if I would think that it's not enough that only these cells switch. Other hits need to happen. The macrophages play still a super important role, and endothelial dysfunction, all the mechanisms that we know, and we see these Tregs switching, we think they contribute to that. Now, if you have a patient where you have plaque rupture, you will probably see, because they had atherosclerosis for such a long time beforehand, the switch of these cells. If the myocardial infarction now itself makes it worse, we don't really know it. And at some point, we see that the majority of cells have switched. So probably it's not known if it's reversible. So we need to also look into the epigenetics of these cells, like how solidly changed are regulators of genes. And yeah, it's a huge effort still to come. And I think it's a very valid question that you have. Yeah.
That's very beautiful science, definitely. It's very beautiful.
Yeah. Thank you very much. I mean, I got to give credit to Klaus, right? I'm taking some bits and pieces with me.
I agree. I'm going to ask you a career advice. So I saw you have really good collaboration, right? So as early career as myself, or like thinking about establish myself in the field, how did you manage to reach out to your collaborators and get them involved in your science?
At the local site, it was relatively, let's say, unproblematic. I just cold emailed them and said like, hey, I'm, the new one here. I have this in mind. Are you interested? And then they usually come back and say like, yeah, fantastic, come by, let's do a meeting. And then we talk. And this collaboration with the pediatrics department is super fruitful. We are starting to enroll patients, I hope very soon. The ethics is almost done for submission, let's put it like that. And yeah, I mean, we always know these ethical agreements are an absolute nightmare to get approved, but I think this is how I went over it. Here at campus, I'm still reaching out to a lot of people that I just think they do cool stuff. And then you get into talking. So for example, I also contacted a person at the Max Planck Institute for Metabolism Research next door. And I wanted to chat with her about something else. And I took a project from Klaus's lab with me about the olfactory receptors in atherosclerosis. And she was looking at me, no way, you're also interested in olfactory receptors. I'm looking at them in obesity. And then you start a completely different conversation all of a sudden, where you actually wanted to talk about something completely different. I think that's just happening, that everyone has a lot of projects, and then all of a sudden you talk about something very different. But yeah, reaching out, cold emailing has worked for me so far. Really, really great. And asking then already people that you're closely working with. So my lab is embedded in a cardiology department here. And I'm working closely with the chief of cardiology and his lab manager. And then I asked them for advice. Hey, I would like to do that. Do you have someone to recommend me? And then they introduce you. So it still works over the established people also very well.
Sounds good. It's like reaching out and asking put yourself out
there. Exactly. I can only say, don't be shy or afraid. Just, you know, send out an email. And from my experience so far, everyone is super nice and more than happy to help you, to get you started, give you information that is sometimes even not in the papers, and then go like, hey, yeah, of course, here, let's talk about this. Let's have a phone call. That really helps. And then, of course, it helps to have always, you know, a network of senior mentors that you can rely on. I talk to Norbert almost on a weekly basis. Esther once in a while, we send emails, Klaus once in a while, and Lynn as well. So of course, that's kind of like not super helpful advice, but yeah, rely on the network that you're establishing over time, and the network will always help you and point directions.
That's very helpful.
Yeah.
Does anybody else have questions for Holger? Kind of think we took all your time. Michael.
Could I ask one question?
Absolutely, Michael.
I liked your data with the Treg switching phenotypes. And so since you use SmartSeq2, I was wondering if you thought about trying to analyze RNA velocity to show this switch towards these more pathogenic T cells, or if it was possible for you to even try this RNA velocity.
Yeah, it's a great idea, and we are going to do it once we expand it on more cells. I hope I did not undersell or oversell, but we have so far only a few cells where we are looking at. There's more data hot from the pipeline that Yanal is analyzing, and although Esther was enthusiastically introducing me as a bioinformatics expert, I'm by no means a bioinformatics expert. So SmartSeq2 analysis is getting way more complicated than what you can do with 10x and the nice Surratt vignettes that help you to do things like this. But yeah, this is for the paper, once we have a higher end, something we certainly want to look into. Great point, yeah.
Anybody else have a question? Okay, I can ask the one that I didn't ask before. I just wanted to touch upon what you brought up at the end with the children. Because yeah, if we think about, you know, Apoe knock-on mice or LDLR mice fed a Western diet, or people, you know, it's so complicated, right? It's not only high cholesterol, it's also high triglycerides and high fat and like a lot of things that are going on. And do you have any ideas how to start parsing that out?
No, frankly not, to be honest. So we are collaborating with the experts on that, and we trust their judgment. But I think it's too difficult to really rule it out in humans already with that human diversity. So we are happy to even get samples where we can do some studies on and also see what happens after interventions. So I hope this study will develop as we, hope it goes, and get some really cool data out of it. The diet that an individual is, of course, consuming, that's not part of the study. That's hard to include and track. And then also control for it. Yeah.
We in the lab we're using, because we are trying to parse all these different factors out. We're using, different models, different diets to do or start to look at those things. So for instance, like a low fat, high cholesterol diet. So you can just increase the cholesterol, but not increase so much, triglycerides. The mice don't get obese, right? So maybe trying to incorporate some of these things.
Pardon, were you talking about mice or humans?
I mean, of course, humans, like much harder, right?
Yeah.
But maybe in like starting in mice to model that would be easier.
Absolutely. Yeah. I mean, that is something that we really also want to look into. A lot of these obesity mice or the OB mice have already a deficiency in a leptin receptor, right? And that skews your T cells as well. So I'm not sure what we get.
Yeah, they don't have very good.
Yeah, so
it's not a very good model, the OB.
Yeah. So that is already a little problematic if you have already from the get-go changes in your T cell compartment.
Yeah, but I mean, LDLRs become obese.
Yeah, that's true. I know that some people are already looking into this. So I, of course, want to leave, this to them, and then we do something else about in that line of research. But yeah, it's absolutely valid. We still don't know what the switch does, or what is the trigger of the switch, or what are the triggers of the switch. And I think one of them is hypercholesterolemia. But how does hypercholesterolemia do that? I mean, it comes along with low-grade chronic inflammation. In the plaques, you will face hypoxia. Hypoxia can also destabilize some of the T cells, of course, not in the lymph node, but at least for the ones in the aorta. And then you probably will see site-specific local microenvironments, and it's what everyone always says, but overall it's, I guess, true that the microenvironment will influence the immune response and how it will go. And the systemics will do in the add-ons to it.
Yeah, because if you do a time response of the appearance of those T cells, right, or the switch of the T cells, what time are they increasing? It seems like they're increasing quite a long time after the mice are put on a high-fat diet, or quite a long time after they have high cholesterol, right?
Yeah.
So it seems like it's not the high cholesterol per se, right?
I would not say that overall, because what I did not show, but what is in the paper is that the overall number of those cells actually decreases, of the antigen-specific cells. So overall, they decrease in time, but increase their pathogenic features. So I think they're just overwhelmed by being bombarded all the time with ApoB peptides that at one point they're exhausted, chronically overactivated, and maybe succumb to the environment. And these faint mapper mice that we have there is the generation 1 during my postdoc. So that is also more work to come. We have a faint mapper 2 mouse where we tamoxifen inducibly induce a population of Tregs at the age of 8 weeks and follow them. And you can nicely see the longer you wait, the more cells you get switched. So obviously, at the age of 12 weeks in an ApoE mouse, not many have switched. 16, a few more, 20, a few more, 24, even more. So more and more of these cells are switching. So we're working this up as well, and really want to put the timeline a little bit into it. Yeah. So yeah, it could be aging as well that plays a role here, and it cannot be excluded. And we see that in a high-fat diet, though, at least in Western, so in ApoE knockouts versus Black 6 early on in age, they are there, but they are already a little skewed in their phenotype. That's what I've been showing in the beginning, 8 weeks versus 20 weeks, right? And the FOXP3 raw gamma T cells are already present in ApoE deficient mice. So that insult on the cholesterol metabolism seems to affect at least parts of it early on. And then parts of it can be the continuing low-grade chronic inflammation, and parts of it, in addition, can be aging. Absolutely, yeah, cannot be ruled out.
Are you going to look at regression?
No, we have not yet.
Yeah, because we had that paper. The Moore Lab had that paper, this year.
I saw Sonia's paper, right, with the Tregs switching. Tregs,
Monica's, yeah.
Yeah. Sorry, Monica, not Sonia. Yeah, yeah. And the reason why I'm confusing them both, unfortunately, is we had a colleague at the La Jolla Institute who has the same last name, so I was just mixing up names. My apologies. Yeah, no, that was a fantastic story, and I would not be surprised if you would see the same thing happening in antigen-specific T cells. You see it.
I wonder if those Tregs that they're depleting, during regression are the ones that are accumulating in the plaques, are the ones that you're seeing accumulating in the plaques during regression, and those are important for regression.
Yeah. I mean, we could look at if there are transcriptomes from these cells. We could try to map them together and see if that first of all works, and then actually do some clustering. That would be very interesting.
Yeah, I think there is single cell data from in that paper. It is,
right?
Yeah. Mm-hmm.
Yeah, it's a great point, and we should look into that.
Okay. Any other questions? That was a very long networking.
No, it was fun. I really like it. I mean, it's a complicated topic, and I really enjoyed that there's so many questions and people are interested. So it was really, really nice.
Okay, so I guess we'll wrap it up. Let you have your Friday night dinner and drinks.
Yeah, maybe one beer.
We're meeting tomorrow. We are meeting tomorrow. No, I'm just kidding.
I was already like, Mabruka, don't shock me, please.
All right, guys. Have a nice weekend. Thank you so much, Holger. That was fantastic.
Yeah, sure. Thanks a lot. Good to see you. Have a nice weekend. And well, see you in 2 weeks again.
Yes.
Last session after 2 weeks.